Have you heard of schistosomiasis? Simply put, schistosomiasis is a disease caused by the schistosome parasite. In fact, it has the second largest socioeconomic impact due to a parasitic disease, second only to malaria. Over 200 million people are infected with this parasite, which makes it a huge deal. That's basically 2 out of every 3 people in the United States. Schistosomiasis is one of around 20 neglected tropical diseases, which are diseases that primarily affect low-income populations in areas such as Africa, Latin America, and Asia. These diseases result in a heavy burden to economic development and generally lack sustainable control methods. Schistosomiasis is transmitted through a vector known as a ram's horn snail. Specifically, our lab researches strains of the species Bionfilaria glabrata, or BG for short. These snails are hosts to blood flukes, known to us as schistosomes. So what do we do in our lab? According to Dr. Castillo, we concentrate in efforts to better understand the innate immune system using comparative methods to study interspecies associations. Controlling the spread of the disease is important for the development of the impacted countries. I'm currently working on histological skills involving staining, cryosectioning, learning the anatomy of the snail, and other lab procedures. The goal of this is to become familiar enough with the anatomy of the snail so that I can understand where certain proteins are coming from. Understanding where these proteins are located may indicate higher immune activity in those areas. The specific proteins we're looking for are called TEPs. So what are TEPs? In short, TEP, or thioester-containing proteins, are proteins that contain a thioester group. These proteins are found in mosquitoes, fruit flies, snails, humans, and most things in between. These proteins are important because they tag the parasite and tell the snail's immune system, hey, there's something bad going on over here. Soon I'll be using RNA probes on the snail's organs to see just how many TEPs there are. I'll be using a method called in situ hybridization. This method involves tagging certain RNA strands with a molecule that can then be visualized through a fluorescent microscope. This will be a stepping stone for understanding what the parasite is doing at the infected sites. Eventually, I'd like to explore laser microdissection, which uses infrared lasers to target specific cells for analyzing. If we can understand where these proteins are being produced, why they're being produced, we'll be one step closer towards understanding the parasite-host interaction. This could eventually lead to more sustainable management of the disease and reduce the burden on humans. I'd like to thank the Castillo Lab, the UNGAS Lab, New Mexico State University, and the MARC program for helping me with this video. Thanks for watching.